Hello, everyone, and welcome to today's live broadcast, Deep Immune Profiling of Peripheral Blood Mononuclear Cells, PBMCs, with the CODEX platform. My name is Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Akoya Biosciences. As the spatial biology company, Akoya Biosciences' history represents the rapid advancement driving the field of multiplex biomarker analysis and immunofluorescence tissue analysis. Akoya's portfolio includes the CODEX platform for deep immune profiling and biomarker discovery, and the Phenoptics platform with the vector scanners and OPAL panels for translational and clinical research. For more information on our sponsor, please visit akoyabio.com. Now let's get started. I would like to remind everyone that today's event is interactive, and we encourage you to participate by submitting as many questions as you want and any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. Also, please notice you can share this webinar on your personal social media. Just click on that social sharing tab on the left to let your friends and colleagues know about today's live event. Now, if you have any trouble seeing or hearing the presentation, click on that support tab found at the top of your presentation window, or you can report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Just click on that Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain your credits. I would now like to introduce our first presenter, Dr. Oliver Braubach, Senior Application Scientist, Akoya Biosciences. Dr. Braubach received his doctorate from Dalhousie University in 2011. He then completed postdoctoral appointments at Yale and the Korea Institute of Science and Technology. During this time, he used state-of-the-art optical imaging and optogenetic technologies to investigate the structure and function of the nervous system. Oliver's most recent academic appointment was at Cedars-Sinai, where he conducted research in line with his background in optical imaging. It was during that time that Oliver started to use the CODEX system and decided to follow his excitement for highly multiplexed imaging by joining Akoya. For a complete biography on Dr. Braubach, please visit the Biography tab at the top of your screen. Welcome, Dr. Braubach. You may now begin your presentation.
Hello, everybody, and thanks for joining. Today, I'd like to speak to you about the CODEX system, and in particular, what we would like to do is to tell you about the diversity of the applications for this system. The CODEX system was launched in 2019 by Akoya Biosciences. And at that time already, it was awarded an award for one of the top 10 innovations by the Scientist magazine. So we're very proud to present this system to you and speak to you about one particular application today. Deep multiplexing and spatial profiling are on vogue today. We know today that highly multiplexed analysis of tissues is crucial to understanding how an organ works or how a disease progresses or forms. But one of the things that's a little bit more new and that's really being appreciated is the spatial component of it. So you can see here on the bottom left, is a piece of lung cancer that was highly multiplexed imaged with CODEX system. Breast cancer, skin tissues. These types of tissues, any type of tissue really, has many, many biomarkers in it. And it's absolutely crucial to understand what these biomarkers are doing and also where they are. And therefore, a pragmatic solution like the CODEX system is very, very exciting scientific development towards understanding the makeup of these systems, but also the function of these systems, and eventually the dysfunction of these systems. And what we're going to do today is a little bit different. We're actually not going to talk about spatial biology. We're going to talk about using the CODEX systems to image PBMCs. PBMCs are cell suspension. Inherently, don't really have any spatial organization. But what we would like to show you here is that this system is so widely deployable that you can actually analyze cell suspensions with it. So we're very excited about this PBMC application. And with that, I would like to turn it over to Jon Mulholland and Anum Khan, both from Stanford University, who developed the protocol for the PBMC imaging and who wrote it up and will have made it available to the CODEX community at large. So Jon will move on now, and we'll speak about how he has implemented the CODEX system in his imaging core. And Anum will then speak about how the experiment is conducted. I will rejoin later to talk about the data analysis. Thank you.
Thank you, Dr. Braubach. I would now like to introduce our next presenter, Jon Mulholland, Director, Cell Sciences Imaging Facility, Stanford University. Jon trained as a cell biologist and microscopist while working for 15 years in the laboratory of Dr. David Botstein, starting at MIT, then Genentech Incorporated, and finally Stanford University. He has been director of the BECKMAN Center's Cell Sciences Imaging Facility, CSIF, at Stanford University since 2001. During that time, he has overseen significant expansion by establishing its electron microscopy core, expanding components of its confocal and two-photon microscopy services, as well as adding FLIM, super-resolution imaging instrumentation, and lattice light sheet microscopy. In 2019, with support from Stanford School of Medicine and the Stanford Cancer Institute, he established CODEX highly multiplexed imaging services in the facility. For a complete biography on Jon, please visit the biography tab at the top of your screen. Welcome, Jon. You may now begin your presentation.
Well, thank you everyone for joining us today. My part of the webinar will discuss the need for highly multiplexed spatial imaging, introduce the CODEX technology, and then I'll discuss its implementation in our facility here at Stanford. So let me first tell you just a little bit about our facility, the Cell Science Imaging Facility. Our facility has historically been focused on providing high-resolution, multidimensional imaging technologies to researchers both here at Stanford as well as outside of Stanford. This is done in 3 interdependent cores: the fluorescent microscopy core, the electron microscopy core, and our new spatial multiplexing core. It is important to note, I think, that we are not a histology or a pathology lab, and those services are provided by other university cores. So providing highly multiplex spatial imaging of tissue and cell suspensions is a totally new service for us. As you can see in this slide, our spatial multiplexing core offers 2 multiplexing services, the array tomography and the CODEX. Array tomography faces all the known challenges, which we're going to discuss today, of traditional serial immunofluorescence methods. It offers relatively low-plex tissue imaging on plastic embedded tissue that is serially sectioned as an array onto cover glass and then subjected to iterative immunofluorescence imaging. That's 3 antibodies at a time plus a DAPI channel. Array tomography sections, though, can be cut as thin as 40 to 50 nanometers and thus can achieve super resolution in the Z dimension, which makes this technique a little bit unique. On the other hand, CODEX, which we have now been working with for about a year or so, avoids many of the challenges faced by array tomography and other traditional serial IMF methods and offers highly multiplexed imaging on a single tissue slice at cellular resolution. So is there a need for multiplexed imaging, and what specifically is that need? This graph, assembled from PubMed searches, shows a steep rise in the number of publications using multiplexed imaging over the last decade. So clearly, multiplexed imaging is a growing technique and is meeting an increasing need. But what is our need as researchers for multiplexing, and more specifically high or even ultra-high multiplexing? What we need is to label large numbers of markers over whole tissue slices for spatial coordinate information along with intensity measurements. We need low cost, no spectral overlap, adequate dynamic range, and single cell or better resolution. Importantly, we also need analytical tools comparable to those employed by cell sorting, image mass spec, and other high-plex technologies. So with those applications, we can map epitope presence along with intensity, as shown in figure A here, segment out single cells and assign and cluster functional states based on coincident epitopes, as shown in Figure B, and then, as illustrated in Figure C, explore those functional states in the context of cell neighborhoods and cellular niche. But there are challenges to doing all that with traditional serial immunofluorescence methodologies, as we are all familiar with. Due to issues of species cross-reactivity and spectral bleed-through, we typically limited to 3 probes per cycle. Then there are problems inherent in removing the sample from the slide, stripping, then reprobing, and again imaging for each successive round of 3 to 4 antibodies. Then, post-image acquisition, there are the issues of overlaying and aligning of images for each successive round of probing and imaging. One solution to avoid the problems inherent in immunofluorescent methods altogether is to instead use mass-based metal reporters, such as in conjunction with imaging mass cytometry and multiplexed ion beam imaging, better known as MIBI. This allows for potentially greater than 100 probes per assay, straightforward panel design, no autofluorescence, a dynamic range order of magnitudes over immunofluorescence. No stripping and high resolution is achievable depending on the beam diameter. But this requires expensive equipment and reagents, and complicated technology requiring a high degree of expertise to operate. Additionally, there's sample destruction. And finally, there just isn't many accessible instruments available, particularly in shared core facilities. A more familiar approach, which requires far less expensive instrumentation and expertise and costs far less, is to use the CODEX approach. CODEX was developed several years ago in Gary Nolan's lab, as Oliver has pointed out, and as you know, that technology has since been commercialized by Akoya. Similar to traditional serial immunofluorescent techniques, CODEX uses iterative rounds of staining and imaging, as you can see schematically here. But in contrast, the primary antibodies are applied to the tissue slice altogether as a panel in a single step, and then the tissue is repeatedly probed and imaged with reporters, as demonstrated here. So how does this work? Well, specifically, let's take a look at the chemistry. The basic chemistry of CODEX is relatively simple. Each antibody in an antibody panel, which can be up to 40 antibodies, is conjugated to a unique oligonucleotide barcode. The entire antibody panel is then applied to the tissue in a single step. The stained tissue slide is then mounted on the microscope's CODEX microfluidics platform. After that, the fluorofluor-coupled oligo reporters that are complementary to the antibody oligos barcode are applied, 3 colors at a time. And the 3 individual fluorescent channels are then imaged as indicated in this slide. So again, here we see the CODEX workflow where the antibody panel stains the tissue all in one step. The tissue now on the microscope is then probed with a set of 3 complementary oligo reporters to reveal specific markers. The tissue is then imaged, 3 reported channels at a time, plus DAPI. Here we see just the first cycle. Then still on the microscope, the oligo reporters are eluted, and we repeat the workflow in sets of 3 unique reporters plus DAPI. Importantly, this is all automated on the microscope. There's no removal of the slide until the last round of imaging is complete. Here we're showing 16 plus cycles. So what are the advantages of CODEX over other highly multiplied methodologies? Well, from our experience in our core, there has been no restriction on antibody host species, so panel design is flexible and simple. The 4-channel sequential imaging produces little to no spectral bleed-through and is done on your familiar immunofluorescent microscope. So there's no learning of new technologies, really. The platform is automated, so after the tissue is labeled with a panel of antibodies and mounted on the CODEX, you can pretty much walk away. When needed, we can achieve diffraction-limited resolution for subcellular localization as well. And importantly to us, the data is in a familiar and third-party compatible file format, the .fcs. So we see CODEX. So we see in the facility CODEX as an affordable basic science tool for our core. But it's important to note it's not a high-throughput tool, and there's other instrumentation out there in the market for that. Okay, so in this slide, I'm simply demonstrating the labeling and imaging quality of a 5-plex panel plus DAPI on a tissue microarray. There are good signal-noise ratios in this imaging with high resolution, and this will also be evident in the data that will be presented on the PBMCs. Our next speakers, Anum and Oliver, will also show data from PBMCs with much higher multiplexing. Okay, so on this slide, I'm basically just wanting to show what our startup costs were for starting CODEX within the facility. And as you can see, the total price of approximately $230,000 for startup is a very reasonable cost point for most core facilities. Lastly, in the CSIF, we offer CODEX multiplexing services that include profiling using Akoya's established screened antibodies and conjugates, profiling using Akoya's and community-established validated panels, using custom validated antibody conjugates and panels. And it's important to note that profiling can be done with fresh frozen as well as on FFPE tissue slices. So for more information about our CODEX service and custom conjugation costs, etc., we can be contacted at the URL showed at the bottom there or at my email directly. I'd now like to turn the webinar over to Anum Khan, who will tell you a little bit about deep profiling on PBMCs with CODEX.
Thank you, Jon. Our next presenter is Dr. Anum Khan, Staff Scientist, Cell Sciences Imaging Facility, Stanford University. Dr. Khan came to Stanford after receiving her doctorate from Dartmouth College in 2018. During her graduate training, she used a variety of microscopy techniques to investigate how cytoskeletal proteins called septins assemble in cells and in vitro. She's currently working on optimizing the CODEX service pipeline, as well as collaborating with Stanford researchers on using CODEX to understand various biological problems and disease progression. For a complete biography on Dr. Khan, please visit the Biography tab at the top of your screen. For a complete biography on Dr. Khan, please visit the Biography tab at the top of your screen. Welcome, Dr. Khan. You may now begin your presentation.
Thank you, Christy, for the introduction, and thank you everyone for joining us. As mentioned by Oliver and Jon, CODEX really was developed as a solution to combine ultra-high multiplexing with spatial data needed to understand the tissue architecture. Here we're using CODEX to demonstrate its application on understanding cell suspensions, and we're using PBMCs as our cell suspension models. So single-cell studies done on cell suspensions are very important to understand a variety of different biological functions. So for example, in order to understand a body's response to a foreign pathogen, we can use cell suspensions such as the blood to understand how a body reacts to it. We can also use cell suspensions to identify an unknown microbe, for example. Cell suspensions can also be used to understand the underlying cell functions such as phagocytosis or cell cycle or apoptosis of a cell. One commonly used example of cell suspension in a clinical setup are tumor biopsies. So in a cartoon shown on the left over here, you can see that tumors at various stages are a heterologous population of different tumor cells. So in order to understand how the tumor progresses, we need to be able to do highly multiplex studies on these tumor biopsies to look at the entire population of cells that are present within the different stages and understand disease progression. Another commonly used example of cell suspensions in a clinical setup is blood, shown on the right over here in the schematic. So for example, in a situation such as the one we're facing with the highly infectious disease such as COVID-19 these days, we can use blood to understand the body's immune response to something infectious such as the coronavirus. So cell suspensions give us an insight into how the body is reacting. We can also use blood to understand or to look at different circulating molecules that the body produces in response to these viruses, for example. So Here, we're using CODEX as a platform to understand or to look at one of these cell suspensions. So what we did, I'm briefly going to walk you through what we did for this experiment. So we took human PBMCs. They were fixed in solution using 1.6% PFA. They were then added to a polylysine-coated cover slip. They were then stained on the cover slip for 3 hours at room temperature with 26 different antibodies. The excess antibodies were washed off, the cells were fixed again, and then were imaged using the CODEX platform. If somebody is interested in replicating this protocol in their own labs, we do have a write-up. I'm not going to go into the detail of each of the steps, but I just wanted to show you briefly what it looks like, and if anyone's interested in replicating this or getting access to this protocol, you can either contact me or Akoya at the 2 email addresses shown at the bottom of this slide over here. So in order to do highly multiplexing to demonstrate high multiplexing on the cell suspensions, we use a 26 antibody panel. So for this purposes, we used Akoya's validated antibody panel that they have used for fresh frozen tissue. This includes a repertoire of different markers for immune cells, including T cells, B cells, monocytes, etc. We also included 2 negative markers as controls, highlighted in red over here. And on the right, I'm just showing the 7 of the 26 markers stained on the right-hand side in this image. To just show you what these images and the staining pattern look like, I'm going to show you some of the markers within this panel in the next slide over here. So you can see a field of view in this slide, and I'm going to walk you through some of the different markers that we use. I'm not going to go into details, but just to show you what the staining pattern looks like, and then we see a strong signal to noise. I'm going to just walk you through the different images we see in some of the different markers. So before I hand it over to Oliver to dive into the data analysis part of the study, we wanted to perform some basic quality control measures to assess CODEX performance on these PBMCs. So it's not a given that these cells are going to remain adhered onto the cover slip, since the CODEX involves multiple washing steps over multiple cycles. It is a possibility that some of these cells can get washed away. So on the left here, what I'm showing you is a series of images taken over multiple cycles. So you can see the cycle number going from 1 to 12, and then you're seeing the same nuclei over these multiple cycles. Some of the flicker you see is because of the frame shift, but we don't see any significant loss or increase in number of nuclei that we're seeing in this frame. On the right here, we've quantified the number of nuclei we're seeing over 2 different experiments, and then 4 different imaging areas. So we're looking at a total of about 80,000 cells. And on the y-axis, we have the number of nuclei that remain over a 12-cycle experiment. And you can see in the red line shown over here that for most of the cycles, the line remains flat. Around 100% of the nuclei stay over the duration of the entire experiment. We do see some jitter around cycle 5, but we believe it is because of some of these frame shifts more so than any significant loss of cells. So what we conclude from here is that there's minimal loss of cells fixed onto these polylysine-coated cover slips over the duration of a multi-cycle CODEX experiment. Another thing I wanted to show you was what kind of cell densities we can get from such experiments. So here I'm showing you 2 different experiments, 2 different densities. So on the left is the low density area, on the right is the high density area, so about 1600 cells per millimeter square, and on the right is twice that much. And just to point out, these are single view images. So assuming the same density throughout, we can calculate for a total imageable area of 15 by 15 millimeter. You can get somewhere around almost 400 or 360,000 cells in a low density area, such as this one on the left, and around 700,000 cells for a highly dense area, such as the one on the right. It really depends on what numbers you're looking at, but we think both of these densities are okay for any single cell studies. So, and then if we zoom into one of these highly aggregated nuclei, what you can see here on the left is that we do see some debris, and this is just part of the experiment. There's not much we can do in terms of getting rid of the debris, but one thing that can be done post-acquisition is that you could draw a ROI to exclude areas that include that debris. Another thing that we really need to do is to be able to segment out these cells to be able to count them. Oliver's going to go into detail, but I just wanted to show that even in high density areas, such as the one shown on the right, in these highly clustered cells, our CODEX algorithm that Oliver's going to talk about further does a good job of segregating or segmenting these nuclei into individual cells. So before I hand it over to Oliver, I just wanted to highlight some of the advantages that one may have by using CODEX for studying cell suspension as opposed to something that is more commonly used, such as a flow cytometry technique. So for most commonly used flow cytometers, you can do up to 4 to 8 different markers, whereas CODEX allows you to do very deep or ultra-high multiplexing, almost 10 times that of the flow cytometer, while letting you do up to 40+ markers at any given time. The second advantage is with flow cytometer, there is a large number of controls that are involved. So for example, some of these controls are, you need to do unstained controls for each of the markers that you're doing, fluorescence -1 controls. These are just only some of them. There are a lot more. With the CODEX, on the other hand, you can get away with a limited number of controls. And this is because, as opposed to a flow cytometer, in a CODEX, you do have the ability to visualize your cell morphology, as well as to look at the localization of a certain marker within the cell. So with that, I'm going to hand it over to Oliver, who's going to go into further detail of the analysis that was done on PBMC. Thank you.
Okay, thank you, Anum, and also thank you, Jon, for the great presentations, and especially for making this experiment happen. I think this is super exciting, and I'm going to talk to you about some analysis that we did at Akoya with the data that Jon and Anum have provided. So first off, I would like to tell you that we have an end-to-end software solution to analyze these data, and everything that you will see today coming out of me at the end of this seminar will be done with software that's provided with CODEX. So we have a CODEX instrument manager. This is what you use to control your microscope, your light source, set up the tiling, essentially to acquire the imaging data that is collected as part of a CODEX experiment. Then we have a CODEX processor. This is a really big part, a really important part for any lab that, for example, doesn't have access to a bioinformatician, because this processor will do drift compensation, background subtraction, deconvolution, cell segmentation, a lot of functions that are pretty nifty and very, very useful. And then lastly, we have the CODEX Multiplex Analysis Viewer, or MAV, which is the platform that we use to do the actual data analysis. And that is a platform that I use to generate all of the data that I will show you now. This software is based on ImageJ plugin, so it's available right now. You can download it, you can install it on your computer, all for free. And it's very good, actually. It combines imaging data with some very higher level functions like gating, t-SNE plotting, k-means clustering, spatial interaction analyses. All of this can be done inside of the MAV. And I'm going to show you some of these outputs right now. So as Anum had said, the principle of our analysis, the way we get to converting imaging to numerical data is via cell segmentation. So here you can see a bunch of nuclei, and here is the actual segmented output. This is a rather dense region of your cover slip. You can see it does a pretty good job. The output here in tabular form, you can see every cell has an ID. In this particular case, there were 35,000 cells, and then every cell has a multiplex labeling pattern, shown by AB1, that's an antibody 1, and so on and so forth. So antibodies can be labeling either very strongly, which is here, you can see 48,000, that would be a pretty strong signal, versus very weakly, which is 394, which was essentially a blank. And if you think about it, in this particular example, you would have 36 antibodies over 35,000 cells. It's an incredibly complex grid of data. So what we have is we have an FCS and CSV output, that is readily generated by MAV. So for those of you who are bioinformaticians in your lab or are bioinformaticians, you can take these files and put them in any platform that you like. For those of you who would use our software for their data analysis, you'll be working with MAV, and I'm going to show you once again some of the things that you can do with this. So first off, I would like to show you that we have some very nice built-in quality control functions. So you can see this here. This is blue square here, this blue rectangle actually, sorry. This shows you imaging data from 2 regions in one experiment. Each one of these histograms works as follows. You have from left to right the labeling intensity or the fluorescent intensity of the CD45 antibody. So the further over to the right it is, the more intensely it labels. Over on the left side, it's a blank. On the y-axis, you have the frequency of cells that are located anywhere along this intensity spectrum. So for CD45, for example, you can see there are very few cells sitting around on the left edge. That means there's very few weakly labeled cells. Whereas over here, you have high frequency of cells that are labeled very nicely with CD45. This is precisely the result you want to see in a quality control for PBMCs because by definition, these cells should be CD4 positive. But what we would like to show you, especially here, is that this gives you sort of a snapshot overview of your data quality. And for that, let's focus on the second column here. So in the second column, you have CD3 labeling. CD3 labels T cells. In this particular experiment up here, 2 different regions, you see this sort of dual spike histogram. Lots of cells in the low intensity regions, lots of cells in the high intensity regions. These will be your blanks. This is what you will cut out, as it will show you in your second. And these are your actual positive staining results. Now, if you go down to experiment number 2, you can see just a single skinny peak. It's all hovering around the lower intensity region. So you would know right off the bat, there could be something wrong with this experiment. Maybe the antibody didn't label properly, or maybe the antibody that the user has chosen in this particular regard is not ideal for the particular application. This is one thing I would like to bring up here is because, again, and we like to reiterate this, the user can choose the antibody that they would like to use. So if this antibody for this particular application is not ideal, you can go back. You can find a better CD3 antibody, reconjugate it, and use it for your CODEX experiment. So this quality control is very good. Get a first look of your data. If you're happy with your data, you go on and you actually do some analysis. Within MAV, we have built-in gating functions, and they allow you to sort your data rather seamlessly. So if you have used flow cytometry currently or in the past, you will probably know what this is. This is a scatter plot in which you can see background fluorescence on your x-axis and CD3 fluorescence on your y-axis. Each dot is a cell. If you consider this small little dot here, you can see it's very high in background fluorescence, very low in CD3 fluorescence. It's probably some type of debris. If you look at this dot over here, very high in CD3 fluorescence, very low in background fluorescence. That's probably a very clean T cell. So what we do, and what you do, for example, in flow cytometry, is you do gating. You really draw a manual gate around a whole bunch of these cells, and then you determine whether or not these are indeed cells. So with MAV, with CODEX, this is very easy to do because you can draw a gate and you get instant visual feedback. So here you can see this black gate. I just chose a small region of interest in the image viewer, and you can see each one of these is a T cell. Likewise, if you draw this yellow gate, you can see here, there are a whole bunch of dots in here. They could be cells. You don't really know. Until you have visual feedback, you can actually see that these are all false positives. So this is one of the things that I particularly believe are the strengths of doing this kind of an experiment with MAV, is because you get instant feedback. You can see this is a true signal, this is a false positive. This is immensely useful, very interactive, a very exciting way to actually do gating. So now you've identified your CD3 cells, you can gate these particular cells once more, and you can interrogate, and you can say, which one of my CD3 cells contains CD4? Tregs, a subtype of T cell. Once again, you get a distinct distribution in your scatter plot, you draw your gate, and you will get visual confirmation of, say, these red cells. These red cells are your CD3, CD4 Tregs. Do this again. You can do the same thing with CD8. Draw another gate, Again, you're looking at these CD3 cells, and then you can see all of the different cell types together. Once again, you have visual confirmation, and it's a very interactive tool to work with. Now, what I would like to show you in this next slide is a proof of principle, where we basically show you that this experiment works as it should work based on reference values. Up here, you can see 5 different types of cells. You have 3 T cell types, CD3, CD4, CD8 positive ones. There's a B cell type, and there are stem cells. Each one is shown in a different color. Down below are histograms. Now, these histograms have 2 data points in them. First of all, there's a dashed line. The dashed line is your reference value. That's what we would expect these cells to be like from clinical reference guides and from the Atlas of Hemopathology. That's what we use to determine these values. So this is where you would want your T cell proportions to be. Each one of these dots, black or colored, is an experiment that we did using the CODEX system with PBMCs. You can see that almost every experiment falls within the range. So this is a very nice confirmation that your CODEX data are consistent with reference values, and that indeed you can use this experiment to analyze PBMC immune profiles. Now, one thing I would like to point out, just because we're talking about data and different tools, if you look at the CD8 result, you can actually see that there's one experiment that falls outside of the expected range. So you could red flag this experiment. You could say, oh, there's something wrong with the patient, or all kinds of ideas that you can get out of this. But remember, we have all kinds of different types of data available. In this case, you can go back to your CD8 quality control data, and you can look at the histograms. You can look at the frequency distributions of your intensity signals. Here you will recognize that for these top 2 dots, you will have this bimodal intensity distribution. Now, especially here, you have a lot of CD8 cells that have high intensity. If you look at the second experiment, it's just this one peak that's sort of mid-range to low intensity fluorescent CD8 staining. So I, as a researcher, would now conclude, well, maybe this was not ideal staining. Maybe this antibody was not the right one. Something is wrong with this experiment, rather than there being something wrong with the patient. So this is an example of not only the tools that you have, but that you can use our tool, CODEX system, to analyze PBMCs. So now I'm going to switch gears a little bit, and I'm going to go into the really deep multiplexing aspect of this. Anum has already shown this slide, where essentially here you can see a whole bunch of different PBMCs with 5 different labels. Let me switch over. You see same field of view, but different PBMCs with another 6 labels. Here are another 7 labels. Here are another 5 labels. All told, this pattern, this result, these data are very, very complex. You have 20,000 cells. You have 26 unique reported readouts. If you were going to manually gate all of these cells, manually analyze all of these cells, it would take you hours, days, weeks, if not months. It's very difficult to do this manually. So what we'd like to show you now is that you can actually do all of this via k-means clustering and unsupervised organization of your data in MAV. So this function, this clustering of data into distinct groups, into distinct phenotypic proportions can be done within the MAV software. And that is what this looks like when you do it. So what you can see here is a result in which I've done unbiased k-means clustering for one sample, sample number 1. You can see on the left-hand side is a heat map. This heat map summarizes the antibody labeling for each one of the 26 antibodies. This is the result that Anum had created for us. I've done the analysis for it. So every antibody is listed down here, and then there are cell types here. The heat map is coded where red is hot. That means an antibody stains strongly. Blue is cold. Antibody stains weakly. So the clustering algorithm has identified, I think, 14 different cell types. Once that happens, you have to manually curate your list. You have to go through the list. You have to annotate your cell types based on what you know from the literature. So we know that a B cell, for example, labels with CD19. You can see here, 2 cell types are labeled with CD19. Those are your B cells. The same is for your T cells, for your precursor cells, and for your circulating epithelial cells. The manual curation involves a little bit of filtering your data and cleaning it up because clustering algorithms tend to overdo it. So they will always identify more types of cells that really are there. So the user has to know or has to be somewhat familiar with the biology of the system, because you do have to curate your data. But all told, we have about 12 different types of cells on this heat map. You can see here the numbers of these cells. They're shown in this histogram. Blue, B cells, green, you have Tregs, and in this color here, you have macrophage precursors and other cells unassigned in black. To the right-hand side, you have a t-SNE plot. The t-SNE plot basically shows you every single cell as being a little dot. Here's an example of a single cell. And these dots are then grouped by color and position into distinct phenotypic populations. So you can see the color coding of this t-SNE plot correlates with this histogram, and these are the names of the cells. You can see the Tregs are the most prominent population in this PBMC sample, as are these GM precursors. So the question now is, this is exciting, this is good. You've identified 12 different phenotypes based on unbiased clustering. This is really good because it's a relatively hands-off approach, and it is very deep immune profiling. So now let's ask, how consistent is this result? And I'm now going to switch my slide over, and you will probably barely notice a difference because these results are really similar. So here we go. The slide switches over now. You can see, and I'll go back just to show you, the heat maps, they change a little bit, but not drastically. And that's really nice because it shows you that the clustering algorithm, now once again, we're doing unbiased k-means clustering for sample number 2, is very consistent. This time, we're finding 11 cell types. So we're actually missing one cell type that we had found before, but everything else is very much in line with it. We got our B cells, our T cells, we got our precursor cells, and others. This is the t-SNE plot. Now I'm going to put these two data side by side, and you can see this a little bit better. So what I would like to show you on this slide is the consistency of clustering data. Again, we're speaking about deep phenotyping, deep immune cell phenotyping in a PBMC sample. Hasn't been tried before. This is a new experiment, so consistency is key. So here you can see two t-SNE plots. You got your sample 1 on the top, sample 2 on the bottom. Now with clustering and these kinds of representations, every result will look different. You'll never achieve the same geography. For example, this green area here is here in this particular plot. It's over here in this particular plot. That's the nature of how these results are generated. What's important to recognize is that you have the approximate shapes and sizes of these patterns are similar. For example, here you have B cells. You can see they're almost exactly the same in the other experiment. I've broken this down in a different way over here on the right-hand side. This is your cell phenotype summary, where we're showing the percentage of the total cell population for each one of these identified immune cell types, color-coded by cell type. You're going from 0% to 100%. And you can see that the breakdown, here color-coded once again, is almost identical. You got lots of Tregs, you got a lot of these precursor cells, and then smaller proportions of CD8 T cells and other types of B cells. So the 11 cell phenotypes that we've now identified in this experiment, they're represented in different clustering results to a very similar degree. They're proportionally consistent, meaning that we find the same amounts of the same different immune cell types across different samples. And this leads us to conclude that CODEX with a MAB analysis produces consistent deep immune profiling of PBMCs. Now I'm going to circle back right to where we began in my last section here, which was the manual analysis of these data. You may remember this slide. This is the output that we got from manually gating immune cells in the PBMC sample. You get your CD8, CD3 cells, CD4 cells, and so on and so forth. These dots are different experiments, and they fall within the reference values that we expect. This is what we've done manually. And I have to say, I did this analysis. It took a fair bit of time. Now, I've overlaid the clustered results on our manual results. So rather than having dots to represent individual experiments, we now have bars. And you can see here M1, that was my manual analysis 1. I gated for a single marker, CD3. M2, I did it again, manual 2. C1 and C2 is my clustered result. But this time, I don't just have information about just one marker, I actually am able to resolve 4 different markers within this CD3 population. The same holds true, for example, if you look over to the right-hand side. Here, I manually gated B cells. I was very happy. They fell within the reference value. Makes sense. Experiment works. Good stuff. But if you look at the clustered result, you can actually see, oh, there are 2 types of B cells now. One is HLA negative, one is HLA positive. I didn't really pay attention to that in the beginning. I wasn't specifically looking for it. But my analysis, the unbiased clustering, led me there right now. And I am now aware of the fact that these 2 cell types are actually here. One last thing I would like to say about this is the manual analysis, the low-plex identification of 4 cell types that you see here took me about the same time as it took me to do the clustered analysis, where I have 12 different cell types. So I think this is a very nice example of deep immune profile PBMCs using CODEX as an experimental platform and MAV as an analytical platform. So to summarize what the 3 of us have talked about today, I'd like to show you this little animation. It looks like Christmas lights, but in reality, what we're doing here, we're doing CODEX imaging of cell suspensions. We're sorting and manually, and we're using unbiased clustering and MAV to really make sense of what these different types of cells are. And we're reconfirming that these findings that we have are in line with what is expected from the literature. So this is a good example of deep immune profiling of PBMCs. Now let's put this into some perspective. Everybody probably knows what this is today. We didn't know this a few months ago, but COVID-19 is everywhere today. And we are all aware of it, and we're all in awe, or maybe scared of this. And it's very, very hot, or very, very important today in science to understand what this virus is doing to human health. So there's a huge push for immune profiling, patient stratification, but also tissue analysis that in samples or in patients that are suffering from this terrible condition. And so we think that there are diverse needs for deep immune profiling. And immune profiling of PBMCs is being done almost on a daily basis now today in view of this condition. And we hope that what we've shown you today will allow you to think about this a little bit more, and hopefully will allow people to use this tool and make some progress towards identifying solutions, not only to this problem, to many others that are associated with our health. So with that, I'd like to thank you for watching on behalf of everyone who participated here. And I would also ask you to join us again on May 12th at the virtual Akoya Academy. Since we're all at home, we're going to keep this going, and we're going to talk about how to analyze PBMC data with MAV. So we're going to do a very much more detailed overview of the analysis that I've shown you today, which buttons to click, which functions to use, so that you can actually replicate this kind of analysis with your own results. So if you want more information about this, go to akoyabio.com/virtual events, and we'll see you then. Thank you very much.
Thank you, Dr. Braubach, and welcome back, Jon and Dr. Khan. We will now start the live Q&A portion of our webinar. Now to our audience, if you have a question you would like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, let's get started. We have so many great questions already coming in. All right, let's start with this one. Is field of view to be kept constant throughout the process?
Hi, thanks for the question. Yeah, so the field of view is kept constant throughout the imaging process. Initially, what the user does, they define their field of view. So field of view being where your lens sees something, but then you can expand your field of view by tiling into adjacent field of views. All of this is registered in 2 and 3D by the computer, by the acquisition software. And we find that there's very little shift of frame throughout all of the imaging cycles that we've done. And we've tried this up to 20 or more cycles. So we find very high consistency in the field of view. especially in 2D. And one last thing to mention here is that for every field of view, we acquire multiple focal planes, which allows us to reconstruct small shifts, small frame shifts into C dimensions as well. Thank you.
Thank you, Dr. Braubach. Our next question. It is my understanding, it is my understanding that only cell surface protein markers can be captured and not proteins within the cells.
Hi there. So I think that your understanding on this is not quite correct. So it's true that most of the markers that we have tested and most of the markers that are currently well established in our experiment are cluster differentiation markers, for example. These would be mainly located on the cell membrane. However, we do know that we can capture proteins and proteins and other molecules inside of the cells. So our system, antibody labeling, penetrates the cell membranes and recognizes intracellular targets and epitopes. So as an example, for this particular experiment, we label Ki67, which is an intracellular molecule. We've also established labeling for other markers such as pCNA and FOXP3. All of these are intracellular or nuclear markers. So it is indeed possible to not only label markers on the cell membrane, but also to recognize intracellular targets. Thanks again.
And thank you. Our next question. Hello, great presentation. This question is for Jon. Is CODEX a single-step staining protocol? And would it be affected by the affinity of the antibodies?
Okay, thank you for the question. Yes, the primary antibodies are all done as a single-step staining protocol. This can be done overnight. If the affinity of the antibody is low, we can do it overnight at 4 degrees if necessary, but it is all done, the primary antibodies are all done in a single step off the microscope. And if you're worried about the number of antibodies going on in steric hindrance, we haven't bumped into that at all yet. So I don't think it will be a problem. So hope that answers the question. Thank you.
Thank you, Jon. Our next question, and Dr. Braubach, I believe this one's for you. Are more narrow wavelength gatings available to reduce the cycling numbers?
This is a good question, and in principle, yes. The answer will be yes, but at the moment, we are using Cy5, ATO 550, and AF 488 as our fluorescent markers. These have relatively broad shoulders or some degree of overlap in their fluorescent emission excitation spectra. So it is true that probably we could increase the cycle or decrease the cycle numbers and increase the load of different fluorescent markers per cycle if we switched our fluorophores. At this point, we haven't done that yet. So we are working with those 3 markers. Remember, this is also a tool for spatial analysis, meaning that our primary user uses this in tissues that are, say, for example, FFPE, formalin fixed. This limits the scope of trying to, load up individual cycles with as many fluorophores as possible because we are dealing with autofluorescence as well. So I think in principle, the answer to your question is yes, but this is a user-by-user decided option. We actually find that most users tend to increase the number of cycles just to get optimal spectral separation of their fluorophores. So this is sort of a bimodal answer here, but really comes down to the user. What we can offer for sure right now is we have Cy5, AF 488, AF 750, and ATO 550, and that's what we have. And so the user can mix and match these fluorophores as they please, depending on whatever application there is. Thank you.
Thank you, Dr. Braubach. Dr. Khan, let's come to you for this next question. These set of proteins detected were all cell surface proteins, correct? And can the system detect proteins that were secreted, such as cytokines?
So for most of the markers that we showed, we didn't go into detail, but most of them were cell surface markers, but we did have intracellular, such as a nuclear marker that Oliver mentioned earlier. We did have a Ki-67. And some of the validated panels, we do have the ability to detect intracellular proteins. However, currently we don't have the ability to detect secreted proteins such as cytokines, and I don't believe we would be able to see them with PBMCs anyways. But no, we don't. We cannot detect cytokines currently.
Thank you, Dr. Khan. Jon, let's come to you for this question. Are there antibody panels for CODEX platform analysis available for species other than human? Canine, feline, bovine, porcine?
Who was that directed to?
Jon, this one's for you. Would you like me to repeat the question?
No, I got it. Currently, no, we don't have any antibody panels for the canine, feline, bovine, or porcine populations, species, but those could be, I guess, done. We'd have to do the conjugations and then do the check the antibodies, etc. But there's no reason not to, as far as I can understand.
We do
Yeah,
have
let me
mouse
chime in here quickly.
Yeah.
Yeah, we have established that the system works from murine tissue. It works with anti-mouse antibodies, and it's just to add here also that we do have custom conjugation of our antibodies. So we have a large antibody catalog. You can see that on our website, and they're broken down by human FFPE, human FF, and mouse. I think mouse fresh frozen. But if a user, for example, is working on non-human primates or porcine models, they can test their own antibodies and conjugate the fluorophores onto these antibodies to see that it works. So any antibody out there is fair game up until the point that you screen it. So you can explore whatever organism you're working with, say zebrafish, C. elegans. We haven't done that. We know human and mouse works, but the application spectrum and the different species that people are working with is expanding. Thank you.
Thank you, Dr. Braubach. Now we have time for a few more questions, but I do want to remind our audience that those questions we are unable to answer live today, and additionally those that come in during the on-demand period, they will be addressed by our speakers via the email address you provided at the time of registration. Okay, let's take a few more questions. Can we use this for three-dimensional imaging?
I think the short answer for that is, oh, sorry. This is
Go ahead.
The short answer for that is no. However, it in theory is possible. This would be to put serial sections on there and then treat each serial section as a region of interest. But you'd have to do the alignments, etc., off outside of the COI software. Yeah,
I'll add to Jon. So the data that we're taking is we do a Z stack and then take the best focus out of that for segmentation, if that's what you meant by 3D data. So there is a Z component to it, but we do flatten it out in the end for further analysis, but we use the Z stack to pick out the best focal plane.
Jon, did we lose your connection? That got a little. Okay, our next question. Does MAB only work with CODEX processor output?
Yes, MAB only works with the CODEX processor output. It was specifically designed to analyze data that come out of CODEX.
Very good. Thank you. And our next question has two parts. To do clustering and phenotype assignment, do we need to purchase an additional module of MAB? And how can we access the MAB tool?
So let's answer these questions in reverse, actually. How can we access the MAF tool? So the MAF, or the Multiplex Analysis Viewer, is actually an ImageJ plugin. You can download that from our website, and you can install that onto your ImageJ platform as a plugin simply by sliding the operation into the plugins folder, and it should work right after. So this is available immediately. You can use it right away, and it's free. The second part of this question is to do the clustering and phenotype assignment. Do we need to purchase an additional module? No, you don't. It's all built into MAF, and the way it comes, the way you download it, install it in the ImageJ, it all runs off that platform. So you have your image viewer with all the built-in tools from ImageJ, which is quite nice. It gives you a lot of different things like ROI selection, level adjustment, and whatnot. Plus, you have these clustering tools, Tisney viewers, K-means clustering. Essentially everything that I showed in the seminar today is built into there. So if you want to, you can set this up today at no cost, really. Thank you.
Thank you, Dr. Braubach. Now let's go with this question. Can you export the data as a .fcs so other tools can be used for data analysis?
Absolutely. So we have all of our data are converted into tabular comma-separated values. So you can export CSV, FCS files, and they can be readily imported into other computational platforms of your choice. A lot of our users are doing that, so this is absolutely no problem.
Dr. Braubach, can you answer this next question for me? How do we sign up for the CODEX May 12th seminar?
So that's this great question. I'll be there to talk to you. So May 12th seminar, all you need to do is go to akoyabio.com/virtual events and look for the Akoya Academy. And you click on that button and you follow through, and there's a registration button that you can click and just type in your name and your contact, and you're good to go.
Very good. Thank you. And let's wrap with this final question. Let's see, we have so many. Actually, you know what? Let's go ahead and take some final comments. I'm so sorry. Let's take this last final question. What markers are the best for characterizing NK cells?
So that's a great question, and I am a CODEX user. I use a lot of different applications. Full disclosure, my background is actually in neuroscience. However, I do know that NK cells are most routinely visualized with CD56. So that's a very standard NK cell marker. I think if you combine it with CD69, you can actually also further interrogate activation states. So we did not actually use CD56 in this particular experiment, but if you type in NK cell flow cytometry, for example, or any type of detection method, generally people tend towards CD56. Thanks.
And thank you. I do want to give our speakers opportunity to say any final comments.
And for me, I just want to say thank you for all our listeners. It's great.
Yeah, thank you for signing up and showing up.
Yeah, absolutely. Thank you all for being here, especially people who are in a very different time zone on a Friday night listening to us speak. It was an honor, and thank you so much for being here. And hopefully you all have a good weekend. Stay safe out there, and most importantly, stay healthy.
Yes, thank you. And before we go, I do want to thank our audience for joining us today also, and for their interesting questions. We had so many questions come in live today that we were not able to answer them all. They will be answered via the email address you provided at the time of registration. I would like to once again thank Jon Mulholland, Dr. Anum Khan, and Dr. Oliver Braubach for their time today and for their important research. We would also like to thank LabRoots and our sponsor, Akoya Biosciences, for underwriting today's educational webcast. This webcast can be viewed on demand, and Labyrinth will alert you via email when it is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. A survey will now pop up on your screen, and we appreciate your feedback. Additionally, when you finish filling out the survey, you will be redirected to the registration page for the upcoming event with Akoya, the Akoya Academy. We hope to see you there. That's all for now, and we thank you for joining us. Stay healthy and have a great day.